So today we're talking about nucleic acids. This is the third organic macromolecule that we're going to be talking about. So first of all, what we need to begin with is what are nucleic acids made of? And they are made of atoms of C, H, and O. That's what makes them organic. But they also contain the elements nitrogen and phosphorus. Okay? For nucleic acids, the subunit or monomer of nucleic acids are known as nucleotides. And this right here is a picture of a nucleotide. Now, a nucleotide is made up of three separate parts. The first part of a nucleotide is going to be your phosphate group, and this is where you see the phosphate. Okay? Remember that the phosphate groups are charged. That's one of our functional groups. This is going to be important later on when we talk about DNA technology and genetic engineering. But as far as the rest of the nucleotide goes, we also have a 5-carbon sugar and a nitrogenous base. So what is going to make a nucleotide or nucleic acid different from another is actually going to be the order of our nitrogenous bases. There's two types of nucleic acids that we need to know of, and that's going to be DNA and RNA. The difference between the two is that DNA contains the sugar deoxyribose, and the structure of the sugar itself is a little different, okay? There is a slight different group right here. The hydroxyl group is a little different in the two. And also one other thing, you might notice that they're numbered 1, 2, 3, 4 on this side, and they're also numbered slightly different on this side, and we're talking about the placement of those hydroxyl groups. And so really, we do want to pay a little bit of attention to the fact that the carbons are numbered. Okay? And we do want to make sure that we know that this nucleic acid, this nucleotide, is used to make both DNA and RNA. Okay? Now, because the shape of these, or the structure of these two sugars, these two 5-carbon sugars are different, this is going to result in a different function for both of those molecules, the resulting molecules. So because the shape is different, and we're talking about the structure of it, okay, we can see a little missing H here, and actually the numbering is also going to be important. So the function is different. Remember, a big concept in biology is that shape relates to function. And of course, this is also going to be due to the bonding pattern. So the shape or structure, and we'll just write to the side, also bonding pattern. Now, one thing we want to go ahead and point out now about the third part of the nucleotide is that for our nitrogenous base, there's actually 5 different nitrogenous bases. So there's going to be 5 forms of the bases. Okay? The bases are going to be separated into 2 main categories known as purines and pyrimidines. Now, purines are a double-ring structure. They are still carbon-based, and they do have some functional groups attached to them. What we need to know is we need to be able to identify the 2 different types of groups of nitrogenous bases, and you also need to remember the specific types of nitrogenous bases that are inside of each group. So for purines, we have adenine and guanine. For pyrimidines, we have cytosine, thymine, and uracil. A way to remember this, first of all, is that for purines, there's 2 different ways that I've heard to remember this, which is AG Ag, if you remember a little basic chemistry, stands for silver. And then you can remember that silver, Ag, is going to be pure. Okay, that's its pure form. Another way to remember this, and this is a little old school, is what I learned was girls are pure. Okay, so G, A, pure, or A, G, pure. So whatever helps you remember it, but you do want to come up with a little way to keep those two separated, the two groups separated. So remember with purines, they are double rings. And this is actually going to matter when we combine two strands of DNA together. With pyrimidines, we do want to make sure we remember that they are single rings. And here there is a little distinction between the two. Here you remember that thymine, the T, is found only in DNA, and the U for uracil is found only in RNA. So for these two structures, DNA and RNA, some differences already that we have is that DNA contains the sugar deoxyribose, that's what the D stands for, and it can also have the base thymine, whereas RNA contains the sugar ribose and can only contain the base uracil, but both of them will have guanine, adenine, and cytosine. The thymine here can't bind to the molecular structure of ribose, okay? So can't bind with ribose. And that's why we have to have the uracil instead. And that's partly because of that chemical bonding pattern of it. The final thing that we want to keep in mind here for this top portion is that we have complementary base pairing that occurs in or with DNA and RNA. So if you remember that for DNA we have A pairs with T, C pairs with G. For RNA we had A pairing with U, there's our little difference, and C still pairs with G, okay? Now, in the naming of the nucleic acids, we have two basic nucleic acids, DNA and RNA. For DNA, we need to remember that this is our double-stranded molecule, so literally two strands of nucleotides. Here you can see one individual nucleotide, okay? What we need to know about DNA itself is that the way that the bonding occurs actually occurs in what's known as an anti-parallel fashion. Okay? Anti-parallel. This means that when a nucleotide is added onto this side, it's actually going to be bonded upward. And then this one, you're actually adding the nucleotides downward, and we'll learn about this 3 to 5 prime process later on. But another thing that we need to notice here is that between our nucleotides, we have these hydrogen bonds, and hydrogen bonds are generally weak bonds, but when there's many of them, that's going to make it stronger. We also need to know that a hydrogen bond is going to be polar, okay? And that's going to be important for later on as well. Another thing that we need to notice here is that you always have this pairing or complementary base pairing of the purines and the pyrimidines, okay? So there's always a purine pairing with a pyrimidine. And that's always, always, always going to occur. And this, because of this, we're actually going to create what's known as a uniform diameter. Because remember, these two strands are going to wind up into a double helix, and then that's what's going to allow it to have that uniform diameter. The next thing that we want to talk about is our different types of RNA. For RNA, this is a ribonucleic acid. It's going to be a single-stranded molecule. And there are several shapes that it can take. The first one, or the first type of RNA, is known as ribosomal RNA. Ribosomal. And also known as rRNA. The next is going to be our transfer RNA, which is known as tRNA. And the last one is going to be known as our messenger RNA, or mRNA. Okay? And all three of these are going to be involved in protein synthesis. And so rRNA is actually going to be the complex in which the amino acids are put together to build the protein. Transfer RNA, if you remember right here, this is an amino acid that's being carried or transported by our transfer RNA, and then the messenger RNA is carrying the code to make or put together that protein. Then there are other types of RNA as well. There's others known as siRNA, and there's just lots and lots more. Okay? But we will get to those later on. But right now, this is what we want to concentrate on. Going on to the next page, we want to look at this central dogma of biology. Okay? So the whole main job, or actually the main idea, okay, central dogma just really means main idea. The main purpose of these nucleic acids is really at the end to make our protein, because proteins are going to provide the structure and help carry out almost all biological activities. So this is your ultimate goal at the very end. But to get to that goal, or to reach that final end product, we need this information, and this is really what you need to remember. All of this is dealing with a transfer of information. Okay, so you start off with your DNA. Notice that DNA can be replicated also, but that's a little side business there. But the information that we're talking about is information used as a code to build our proteins. Okay, that's the main purpose here. And from that, we can take information from the DNA itself, take that code, we will transcribe it, and that's how we get our mRNA, and then we'll eventually translate it, and then we'll be able to get our product at the end. So you do want to make sure that you read through this and look back over this process for protein synthesis, okay? DNA replication, remember, is a separate process. It can happen simultaneously, but that is thinking of a different end result, different product at the end. And one final thing that you want to keep in mind is that that information that's being transferred or that's being transcribed is going to be found in the sequence of our nitrogen bases, because that's really where our nucleotides vary. So a little example of this is that for ATCC, that's going to produce a different protein or different amino acid from GGA. And so these letters, these 4 letters in the DNA and the RNA, technically all 5 total, are going to produce a different amino acid, which eventually is going to lead to a different protein. Make sure that you look over our review questions and that you answer all of the questions that go along with our lecture today.